It's almost as good as sleeping in this class, right? All right, so I hope everybody had a good Halloween. I did. It was a lot of fun. Somebody had more than others, it sounds like. Last time I got talking about different mechanisms of regulating enzymes, and we talked about allosteric mechanisms, which are when a small molecule binds to an enzyme and affects its activity. That can be either positive or negative effect. And I also started to talk a little bit about covalent modification, and I gave you an example of an enzyme that actually participates in covalent modification. And that enzyme was protein kinase A. I said that you will hear a lot more about protein kinase A in the weeks to come. Okay? Protein kinase A itself is regulated allosterically, and it's regulated allosterically in an interesting way. That allosteric regulation of protein kinase A occurs as a result of its interaction with this molecule that you see on the screen called cyclic AMP. Cyclic AMP, as we will talk about later in the term, is a molecule that participates in the phenomenon known as signaling. And signaling is when, just like any signaling that you're doing, it's like a text message to a cell. That text message to a cell tells the cell, do this, don't do that, etc. And we'll see how that information is communicated. But it's communicated through the synthesis of this molecule. The synthesis of this molecule causes this molecule to bind to things like protein kinase A. And when cyclic AMP binds to protein kinase A, it converts the protein kinase A from an inactive form into an active form. And it does it by the mechanism that I briefly discussed last time, and that is shown on the screen. So I want to spend just another minute going over that and making sure that you understand what's happening with this. Okay? Protein kinase A has regulatory subunits and it has catalytic subunits. Okay? We talked about ATCase, which has regulatory subunits and catalytic subunits. And though it has a different number of them than protein kinase A does, the regulatory subunits function, as their name would suggest, to regulate the activity of the enzyme. And the catalytic subunits perform the action of the enzyme. The action of the enzyme, that is the catalysis that is stimulated by the enzyme, is the addition of phosphates to other protein molecules. That's why it's called a protein kinase. I said that the word kinase means adds phosphate to. So a protein kinase is something that adds phosphates onto proteins. Okay? Now, there are a variety of protein kinases that are out there, and most of them are involved in this process of signaling. Okay, we won't talk about any decent percentage of the ones that are actually found inside of cells, but we'll talk about a couple of them during the rest of the term. Now, the addition of phosphate to a protein occurs at specific places on a protein. Okay? The most common places where it occurs is where there are hydroxyl groups in side chains. So this is a joining of the phosphate to the hydroxyl group on the side chain. And so if you think back to the amino acids that have hydroxyls on their side chain, there are three of them: serine, threonine, and tyrosine. And as I mentioned last time, the addition of the phosphate to an amino acid like one of these three that I've just described, the addition of an amino acid to one of these has the effect of changing the charge, because an OH group, of course, has no charge, and a phosphate has a -2 charge. Okay? So what that means then is we've drastically changed the charge of that protein in the location of where this amino acid was, and we might find, not surprisingly, that that change in charge might cause a change in shape, and this change in shape might cause a change in activity. And in fact, that's exactly what happens, and that's exactly why the kinase is very, very important. Okay? Very, very important. So, the effect of adding a phosphate or any covalent modification can be to activate or to inactivate an enzyme, again, depending upon the enzyme. Okay? We'll see examples today of other kinds of covalent, excuse me, other kinds of covalent modifications that can happen, and those modifications will usually activate an inactive enzyme. They will usually activate an inactive enzyme, but this one can have either effect. Okay, well, I want you to keep that in mind because, as I said, I'm going to talk more about the protein kinase A probably next week, and we'll be digging into that in some depth. All right, now, here are some common modifications that can happen to enzymes and other proteins that aren't even enzymes that affects the function and or activity of these different proteins. Phosphorylation is a very common one. Acetylation is a common type of modification. Acetylation involves addition of an acetyl group to a portion of a protein. And in the case of histones, you'll talk about this next term, that can have a very drastic effect on the activation of gene expression. Very positive effect on the activation of gene expression. And there's a variety of others here. And I don't show you this table to get you to memorize a bunch of modifications and so forth. That's not the point. But I want you to realize that there are a variety of covalent modifications that can happen that affect proteins' activity. One of the interesting ones that we talk about, and we unfortunately don't get much of a chance to talk about in this class, is ubiquitination. Ubiquitination is the attachment of a small peptide to a protein called ubiquitin, and that attachment targets the protein that gets the ubiquitin for destruction. So there's actually a flag that says this protein is to be degraded, and it happens because ubiquitin actually gets put onto it. So that's an interesting sort of recycling and control mechanism that cells use to turn over proteins. Okay. The attachment of an acetyl group in the case of lysine, for example, which is a covalent modification, the attachment of an acetyl group also has the effect of changing the charge of a protein, because if you recall at physiological pH, lysine's R group, which is shown partly here, lysine's R group will typically be positively charged. So the attachment of an acetyl group converts that positive charge into something that has no charge. And in the case of gene expression inside of cells, that turns out to be a very important thing. And I just want to spend a minute talking about it. It isn't an enzymatic reaction. That is, we're not affecting histones that, back up and say, the proteins that this happens to are called histones. These are part of our chromosomes. And the histones don't catalyze reactions, but instead they bind to DNA. So when you see something that in a microscope that is a chromosome, that is a combination of proteins called histones and DNA wrapped around that. And the histones have a positive charge, and they have a positive charge largely because of lysines and arginines that they contain abundantly within them. And that turns out to be really useful because DNA is negatively charged. So these guys, these histone proteins, are fairly tiny, and they act like little spools that the DNA can wrap around. And the attraction of the negatively charged DNA for the positively charged histones makes for a very nice, tight complex, which, of course, as I say, leads us to see chromosomes inside of microscopes. If I add acetyl groups to those positive charges on histones, you could imagine that what I might be doing would be loosening the interactions between the DNA and the histones, because I've converted that positive charge, which was part of the positive-negative attraction, into a zero charge, which is not going to be attracted. And in fact, that's exactly what we think happens inside of cells. In order to activate gene expression, the chromosomes, which are all wound up tightly with these histones, have to be loosened so that the DNA sequence can be accessed by the enzymes that make RNA. So this acetylation has a very drastic effect on chromosome structure that allows for gene expression to occur. So covalent modification can play a very, very big role in terms of protein activity. I mentioned briefly what kinases do, and kinases, as I said, put phosphates onto other proteins. The energy for doing this comes from ATP, as you can see above, and you can see the reaction that is involved in putting that phosphate on. Here's the ATP molecule at the very top. Here's the side chain of an amino acid that has a hydroxyl. So this could be a serine, a threonine, or a tyrosine. And by the way, the kinases are generally fairly specific about whether they will put it onto a serine, a threonine, or a tyrosine. Okay? The attachment happens. The product of that reaction is an amino acid now that has a phosphate on it, and the molecule ADP, which is the ATP, which has lost its phosphate. Okay? This is a really good example of something I mentioned very early on. You may not remember the term, but I'll give you the term again here. And that's something we call a post-translational modification. Post-translational modification. So translation is the process of making protein. So something that is post-translational means it's happening after a protein has been made. So we're not modifying amino acids. We're modifying amino acids within proteins. We're not modifying free amino acids. Okay? So post-translational modification is what you see on the screen that's happening right here. And of course, that is a covalent modification because you can see the covalent bond that's occurred right there. Okay. There are a variety of kinases that are out there. Ones that they really break down largely into two big groups. One that predominantly phosphorylates serine/threonine residues. Serine and threonine side chains don't look very different, so many kinases will phosphorylate either a serine or a threonine, or both. And some examples of those are here. Again, I'm not showing you this to get you to memorize something, but just to realize that there are a lot of different kinases that are out there. Okay? I haven't shown you, or I haven't talked briefly, or I haven't talked about the tyrosine kinases. We'll talk about tyrosine kinases later when we talk about signaling. But one of the things we find about tyrosine kinases is they really are a different class of kinase, and they appear to play very major roles in the signaling process, more so than the serine threonine kinases, although they play some roles in signaling as well. Tyrosine kinases frequently will have roles in helping the cell to make the decision, do I divide or not divide? That's a very important decision for a cell to have to make. I like to say that cells are the ultimate control freak. Okay? You might think I'm the ultimate control freak, but cells actually have me beat. All right? Cells are the ultimate control freaks because they need to control as much as they possibly can that occurs within their own world, which is the world inside of the membrane in which they exist. A really good example of that is the fact that if cells have a kinase that puts phosphates on, they also have enzymes that take phosphates off. So putting a phosphate onto a protein communicates a signal, and just like a signal that can't be turned off, imagine that your phone would not stop ringing ever. That would not be very desirable. It seems like what happens in here sometimes. Ha ha. Joke, joke. Okay. But a phone that could not be turned off would be problematic. A signal inside the cell that could not be turned off or that could not be turned on would also be problematic. So if cells have a way of turning a signal on or off, they also have the reverse way of doing it. And in the case of a kinase, the reverse action of a kinase is through something called a phosphatase. Okay? I mentioned this last time. A phosphatase is an enzyme that takes a phosphate off of a protein. Okay? Takes a phosphate off of. So that it has the effect of countering the signal that was done in the kinase reaction that I mentioned briefly. This is a hydrolysis reaction that removes that. You can see the water here, and you can see the free phosphate located over here. Okay. What else should I say here? Leave that alone. Okay. All right. Now, at this point, I want to turn our attention to another kind or another mechanism for regulating enzymes. Maybe before I do that, I should do a limerick. What do you guys say? Let's do a limerick. The tert alcohol was a fool, turned to ketone in making a fuel. The work is complete. The bond gave up heat as it lost all its family jewels. Did I do that before? It seems familiar. I must be losing my touch. Okay. No wonder they can get a giant hilarious laugh. Okay. I picked accidentally 2. All right. I want to spend some time talking about another covalent mechanism that is commonly used inside of cells. Or I shouldn't say, well, it's used inside of cells, but it's more important in the body. And this is what we call zymogens. Okay? Zymogens, as we shall see, are covalent modifications, or back up. Zymogens are inactive proteins that get modified by breaking of peptide bonds. Inactive proteins that get modified by breaking peptide bonds. That modification activates them. Now, this mechanism turns out to be used in the body in a couple of places, and it's used extraordinarily effectively, as we shall see. Okay? Let's consider what happens in our digestive system. Okay? Our digestive system has a need to digest, because it wouldn't be called a digestive system without it. Okay? And that digestion happens as a result of action of enzymes that break things down. Okay? Well, you've already seen some examples of that. You've seen, for example, proteases. Proteases cleave peptide bonds in other proteins. And you might wonder, if I took a protease and I put it in a tube and I let it sit there forever, would it ultimately break itself down? The answer is yes, it would. Okay? So proteases, they don't care where the protein came from. They will ultimately break each other down until there's nothing left that's active in that tube. Proteases, therefore, are very powerful. And proteases, as you might imagine with anything powerful, can cause problems. Okay? So many proteases are secreted by your pancreas, and they are passed into the digestive system. And if you think about it, a cell that's making a protease might have some real problems, because what happens if, when it's making the protease inside the cell starts breaking down other proteins? Could you imagine some problems? Yes, you could. So cells have, or the body has, a way of protecting itself against that. It turns out that when we look at the digestive enzymes, of which the proteases are a very good example, but there are others as well. If we look at the digestive enzymes, they are largely secreted in an inactive form by cells. And that's so that they will not attack the very cells that make them. All right? Those inactive proteins, which of course are called zymogens, as I've noted before, those inactive proteins, okay, pass into the digestive system where there are active proteases that activate them. So this is kind of a cool system. Okay? They're inactive, but other proteases acting on them convert them into an active form. Okay? That's largely how zymogens work. Now, as we will see when I talk later about blood clotting, a similar mechanism exists in blood clotting. The advantage of a system like this is it allows activation of a gigantic amount of protease very, very quickly. Okay? You can imagine your pancreas is secreting some of those proteases right now. And you can imagine that as they travel into the digestive system, they hit a protease which activates them, right? And you can imagine that, will that system start working backwards? Because if it happens too soon, will it back up and back up and back up, ultimately activating proteases as soon as they leave the pancreas and maybe attack the pancreas? The answer is yes, they can, yes, they do. And if that happens to you, you will develop a condition known as pancreatitis. I see a lot of people smiling and looking at me like maybe they've had pancreatitis, although I think if you had it, you probably wouldn't smile. It's a very painful condition. It can be, in some cases, life-threatening. Okay? So activation of a protease at a time and place where it shouldn't be active causes problems for the reasons I've already stated. And so cells, and the body in particular, try to control very carefully when a given zymogen is active. As we will see in blood clotting, that can have disastrous consequences if we activate in the wrong time or wrong place. Okay. So here are some various zymogens that are created. And there's an attempt, it's not very good, but there's an attempt that people made to give these guys a common nomenclature so you could look at something and quickly tell if it was a zymogen or not. And that was the attachment of the OGEN at the end of a name. Okay? So for example, if we talk about the zymogen form of pepsin, pepsin being the active form, the zymogen form would be known as pepsinogen. Same thing with chymotrypsin and chymotrypsinogen, trypsin, trypsinogen, and then we see that people don't always follow the rules. Some of these names were in place before there was a systematic way of looking at that. But generally, if you see pro in front, that will also indicate something as a zymogen. It's an inactive form. Okay. Now, I want to go through the activation of one zymogen, and I'm going to show you some numbers and blah, blah. And the numbers really aren't important, so I don't want you going, okay, it cut at amino acid 13, and I have to know that, and so forth. That's not the point. The point is understanding how a zymogen like trypsinogen can be activated. The activation of trypsinogen occurs in a couple of phases. The first is the attack on chymotrypsinogen by the protease known as trypsin. Trypsin, of course, is the active form, and this active form will be present in the digestive system. Okay? We see what happens in here is that there's a break that is made between amino acids 15 and 16. As I said, you don't need to know the break numbers or anything like that. But the point is that this simple cut right here allows the chymotrypsinogen to be partly activated. The partly activated form of chymotrypsinogen is known as pi-chymotrypsin. And pi-chymotrypsin will act on itself to do some other cuts. So one pi-chymotrypsin will attack another pi-chymotrypsin, and when it does that, it produces cuts that clip out a couple of amino acids. So here, if we cut between 13 and 14, that fragment will disappear. And over here, if we cut between 146, 147, and 148, 149, we'll lose those amino acids there. This is overall a relatively minor set of modifications that are happening, but the effect is to produce a fully active form of chymotrypsin. You'll notice that there's nothing holding these pieces together, but they don't fly apart in the cell. How do you suppose they stay together? Okay, I'll give you a hint. It's a covalent bond. Disulfide bonds. Okay? Disulfide bonds are in place that actually hold this into the right configuration. Now, if we look at what happens to this guy from a structural perspective, what we see right here is the inactive form shown in red and the active form shown in blue, such that the clipping around that amino acid 16 changes this configuration. Okay? It changes from, where am I at here? From this inactive form here to the flipping down where we see blue here. This means that this portion of the protein is no longer sticking out there, and now there's access of substrates into the active site of the enzyme, which is down here. So it's a very common feature of zymogens that the clipping of the peptide bond opens up a region, and that region that opens up frequently allows substrate to come in and bind so that the enzyme can act. In the absence of opening up that region, the active site is blocked, and the enzyme can't catalyze anything. Okay? So it's a very, very common mechanism that happens during the activation of a zymogen. Okay, I'm going through fairly quickly. Let me slow down and ask if you guys have any questions. Yes, over here. How is the first zymogen activated? It's a good question. By the time you're born, you already have the first one activated. So it never starts with 0. Because there's so many in your body, you never have a 0. Yeah, kind of unsettling. Other questions? That's a very common question, though, by the way. Well, you can imagine once you get one active, then you get this sort of cascade of activation of others. Oh, I see a question back here. Yes? Where can you see what? Okay, so the question was, what can you see in this picture that tells you the changes that are happening in the enzyme? Is that what you're saying? Okay, well, what I'm pointing out here is that if we look at the structure in blue and we look at the structure in red, the red being the inactive form, clipping of this bond here basically removes this section so that this now opens up. And so this is we can think of this as a closed door. If we clip that out, if we basically remove the door, then we've opened that up access to the enzyme. Okay? Okay. Now, as I start to say, what we have with these enzymes is a cascade. Activation of one enzyme means one enzyme can go out and activate, let's say, 1,000 other enzymes. And those 1,000 other enzymes can go and activate 1,000,000 other enzymes. This cascade phenomenon is very, very powerful for very quickly making a lot of protein active. In the case of pancreatitis, we can imagine that this thing could get out of control fairly readily, and to some extent it can. So that's why we are very careful when people come down with pancreatitis, because, as I said, in some cases it can actually be life-threatening. Okay, how does the overall thing go, which I think relates to the question over here. Where does it start? Okay. Well, we don't go back to the very first number one catalysis that happens, okay? But this comes pretty close to doing that. What we see happening inside of the digestive system is this cascade that you see right here. It starts with the action of an enzyme called an enteropeptidase. And that enteropeptidase converts trypsinogen into trypsin. Trypsin, you can see, is a very, very powerful enzyme for activating a whole bunch of things. It activates chymotrypsinogen. It activates proelastase into elastase. It activates procarboxypeptidase into carboxypeptidase. And it also activates prolipase into lipase. This guy on the right breaks down fat. Okay? So this trypsin is essentially a central control area for activation of zymogens in the digestive system. And the master switch that turns it on is this enzyme known as enteropeptidase. You also see trypsin points back at itself, meaning trypsin can also activate trypsinogen. So trypsin can make more trypsin. Okay. Now, this becomes very relevant when we talk about the power of proteases. Okay, so I want to give you an example that I think you can relate to from a health perspective. I really like to mention things relevant to human health because they're very important. Okay? Think about the power of a protease. I've already described how a pancreas can be attacked by a protease. Okay? Being able to control proteases is important, and one way of doing it is what you're seeing on the screen. But once you've activated that protease is free to go out and start attacking things. Cells are control freaks. They have other ways of controlling proteases. And one of the ways in which they have of controlling proteases is by the production of things called protease inhibitors. Protease inhibitors. The name, of course, tells you pretty strongly what it does. And if I were to ask you how you thought that a cellular protease inhibitor worked, I would hope that you would sit and you would think about this, and you would come up with the idea that the protease inhibitor is probably something that resembles a substrate that gets bound by the active site of a protease but doesn't get cleaved, thus blocking that protease active site from breaking down other proteases. And that would be a very good answer to that question if I asked you that on an exam. Okay? One such inhibitor is something known as the trypsin inhibitor. The trypsin inhibitor inserts a portion of itself into trypsin and basically stops trypsin from being able to break other proteases. Okay? The trypsin inhibitor works on trypsin very well, but it turns out it's misnamed because we know now that it actually works better on another protease known as elastase. E-L-A-S-T-A-S-E. Elastase is another protease, and it's a protease that's found in a variety of places in our body, one of the places being in our lungs. And you're probably sitting there thinking, why do I need a protease in my lungs? And the answer is, do you breathe? Because if you think what you're breathing in all the time, what are you bringing in with every breath? You're bringing in microorganisms, viruses, you name it, and having some protection in your lungs against those things is very good, right? Proteases provide one level of protection against those things that you're breathing in all of the time. Elastase is one of the proteases that helps to break that, to protect. Now, it's not quite as simple as I just alluded to right here. You might think the more elastase I have, the better off I'm going to be. And you would be very wrong. Because the more active elastase that you have, the more likely you will damage lung tissue because, like I've described before, we have to think about the fact that elastase can just digest proteins. The more it's digesting proteins anywhere, the more likely it's going to attack lung tissue. So the body has a sort of a balance that it does with elastase. It makes elastase in the lungs, and it balances elastase activity with the trypsin protease inhibitor. Okay? The trypsin protease inhibitor, okay, binds to elastase, so the body has a balance of how much inhibitor to make, how much elastase to make, and it might vary with time depending upon the circumstances. During cold and flu season, for example, it might be good to make a little bit more elastase and a little bit less inhibitor. Okay? Now, I mentioned this to you because, as I said, if you make too much active elastase, it will start to attack lung tissue, and elastase attacking lung tissue gives rise to a disease that you don't want to get, and it's known as emphysema. Okay? Attack of lung tissue by elastase gives rise to emphysema. Okay? Now, you can probably start to see where this thing is headed. If you don't have the proper amounts of inhibitor, you're much more likely to develop emphysema because elastase is going to attack your healthy lung tissue. You would be correct. And smokers have reduced amounts of the inhibitor. Why do they have reduced amounts of the inhibitor? Well, the smoke contains reactive oxygen species that oxidize methionine residues. And one of the methionine residues is the binding site of the inhibitor for the elastase. The more you smoke, the more damage you will have to the methionines of your inhibitors, the more active your elastase will be, and the more likely you will have attack and damage to the lung tissue. Okay? So smoking, for many reasons, is obviously not a good thing to do. Here is yet another one that relates to that. Okay. Questions about that? Yes? You can't, as far as I know, you can't inhale the inhibitor. You probably wouldn't want to, because again, you want to have proper balance. And how are you going to balance it properly? So it's not maybe something that you're going to inhale. So, Garrett? Say it again, I'm sorry. Yes? They inhibit proteases. Right, so if a protease inhibitor inhibits a protease, it's going to stop it from cutting, right? Proteases cut proteins by breaking peptide bonds, so maybe I wasn't clear. A protease inhibitor, therefore, will stop a protease from breaking peptide bonds. It will stop the cutting and breaking down of proteins. Yes? Is that like EDTA? No, EDTA is a chelator. That's doing something different. Yes? Would they be competitive inhibitors? Okay, good question. What do you think? Okay, she thinks yes. Other thoughts? How many say they would act like a competitive inhibitor? How many say no? Yes, they would, because they're not binding covalently. So they can come off. So that would act something like a competitive inhibitor. Question over here? Yeah. Okay, so are these affected by pH as well? Any enzyme is in theory affected by pH because that may change charge within a protein. These don't necessarily act like carbonic anhydrase does, so I would say the answer to that is no. So there may be slight effects in pH, but not as dramatic as you will see as carbonic anhydrase. Good question. Somebody else asked me that also. Yes? How do we cure when what? Okay, we don't cure emphysema. People die of emphysema all the time. Yeah. So how do we treat pancreatitis? One way of treating it, you might imagine, would be to dose them with a lot of protease inhibitors, right? That'd be one way of doing that. I'm not a physician, so I'm told not to give medical advice, but you guys wouldn't want to come see me. Yeah, question back here. Can you inhibit an inhibitor? Certainly. Okay. How would you inhibit an inhibitor? Block its binding site. In fact, what you're doing right here is inhibiting the inhibitor. Not in a way that you'd want to. Yeah. Back up here. How do proteases not just eat inhibitors? Well, as I said, they resemble the natural substrate, but they can't be cleaved by the enzymes. So they are blocking access of the substrate to the active site. Okay? So you could imagine that they've got a structure such that they just can't fit into the active site properly and can't be cut, but they do fit into it. So there's no one rule about that. Yeah? Could an inhibitor be cleaved and destroyed by other proteases that they're not designed to inhibit? The answer is yes, they could. So again, we're thinking now of a very dynamic picture that's happening with this because I said body has to balance how much of the inhibitor it's got and how much of the protease it's got. You bet. Good questions. Okay, you guys ready for blood clotting? All right. Blood clotting. Now, blood clotting is the most amazing process, okay? I could take you through and make you memorize every step here, and I don't think that you would actually learn very much about blood clotting. So I'm going to describe blood clotting to you in fairly simple terms. But we're going to have a couple of specific reactions we're going to talk about down here near the bottom that are relevant. Okay? Blood clotting happens as a result of activation of inactive proteins. Okay? So these are zymogens. There's a fibrinogen, there's the ogen, there's the prothrombin, all right? Indicating that these are starting out, they are synthesized as inactive forms. And this turns out to be really, really, really important because imagine you are dependent upon the amount of blood that you have flowing through your system. You cut yourself. If you had no clotting mechanism, which people who have hemophilia have, if you have no clotting mechanism, you will lose all of that blood because your heart will pump it out of your body. Nothing to stop it from flowing out. You take for granted the fact that it doesn't happen, and the reason it doesn't happen is because of an incredible cascade that you see on the screen. The cascade that happens involves amplification of a signal. And at each step, that signal gets bigger and bigger and bigger and louder and louder and louder until we get to the bottom and we've got a ton of active enzyme that ultimately helps to form a clot. It works because normally we have a ton of the inactive form of the clotting material floating around in our bloodstream, ready to be activated. A wound induces physical injury. As we will see, it causes some other things to happen, including the release of calcium ions. And the combination of these things stimulate the activation of that very first enzyme. We can imagine if that very first enzyme gets activated when it shouldn't get activated, we're going to form a clot where we don't want to form a clot. And we all have heard of, or we know somebody who's had a heart attack or who's had a stroke because of a clot that malformed. People who have problems with blood clotting or too much tendency to clot will be put on what's called blood thinner. And that blood thinner is designed to reduce the likelihood of this thing happening undesirably. The wound, however, is a signal to the body that we've got to do something here because if we don't do something here, this person is going to bleed to death. Okay? So having a lot of the precursors flowing in our bloodstream ready to be activated very quickly is important. We form a clot within minutes because if we didn't form a clot within minutes, we would bleed to death. And that clot that we form, we see the visible nature of the scab, but that clot that we form at the molecular level we can't see with our eyes. It forms all on its own, and it forms in minutes, and it's watertight. That is really remarkable. Okay? There are two pathways leading to this activation, and we're not even going to worry about the two pathways. We're going to focus on this bottom part because it's at this bottom part where all the action is. The stimulation of these pathways leads to activation of an important protease, and yes, these are all proteases because in each step, one protease is activating another protease, is activating another protease, is activating another protease, is finally coming down here and activating this protease known as, or the inactive protease known as prothrombin and converting it into the active form known as thrombin. Thrombin, being a protease, activates the last protein in the mechanism, which is known as fibrinogen, converting it into the active form known as fibrin. Fibrin is a self-polymerizing material. It's a self-polymerizing protein that forms an amazing meshwork, a net, if you will. Okay? An amazing network, if you will. All right? This is what fibrinogen looks like. Fibrinogen has 3 different chains known as alpha, beta, and gamma. Okay? Alpha, beta, and gamma. And the inactive form contains these handles labeled with capital letters. So when the capital letter portions are attached, as they are here, you're talking about fibrinogen. And what thrombin does as part of its catalytic activity is it clips those capital letter B's and the capital letter A's off. When that happens, what's left behind on the A will bind to gamma portions, and what's left behind on the B will bind to beta portions. So that we can see how polymerization could actually occur, like here. Okay? We see the betas with, we see the alphas with the gammas right here. What we don't see are the betas in the B, because the betas in the B are projecting out in this 3rd dimension. This provides a polymerizing network that is watertight and that holds everything in place. It's really remarkable. Okay? As this is happening, these interactions that you can see here are not covalent. They're not covalent interactions. However, hydrogen bonds really aren't enough to hold this structure together. Okay? After this structure starts to assemble, there's an enzyme called a transglutaminase that will make covalent bonds between these, and when that happens, you form what's called a hardened clot. And I'm sure you've seen when you cut yourself, at first that clot is fairly fluid. It's easy to bump it and start bleeding again. But after a couple of hours, it's a scab. It's hard. That process of hardening happens as a result of the action of transglutaminase. Okay, now I'm going to go back over that next time. I was going to do a different song, but I actually have a better idea here for you. This is another song that I have, and I'm going to play for you. It's one of my favorites. There's actually a YouTube video for it. Yep. If I can get it set up. And oh, come on. Hold on. What did I do?
Left me breathless.
Can you hear that?
Especially today. An absence from my body makes it hard to say. I'm so horribly upset and reminded evermore. I'll not forget how you took my breath away. I lost a love from smoking camels emphysema care. It seems to me you see those nicotines and tars leave alveolar
That's a friend of mine that died of lung cancer. Okay.